let's get started. I'm happy to see all the interest based on the poll in spatial transcriptomics. And the title of the presentation is The Uses and Limitations of this Powerful Technique. What I'm going to do is give an overview of the methodology. Then we'll get into details of the Nanostring GeoMx platform that we offer at Cellecta, including a couple of use case examples and discussions of best practices in our own service. Mixed into the presentation will be comments about strengths and limitations. Especially during the overview, I'll use some instructive examples from the literature. Along the way, I'll mention some operational details, and that kind of bleeds into strengths and limitations. And I'll finish with a pitch for why we have some advantages in the Cellecta service that we offer. All right, so let's start with a definition. Spatial transcriptomics is an assay that tries to compare patterns of gene expression in cells in specifically targeted areas of interest in tissue sections from tumor or other disease samples. For example, the middle of the tumor versus the edge, near or including immune cells or away from that. And the goal is to understand the genes that drive tumor biology or disease biology, and of course, identify markers for prognosis, metastasis in the case of cancer biology, therapeutics, etc. Alrighty, here we go. Here's another poll question. And Sonia, can you take this one away, please?
Certainly, Lester. I'm going to launch poll question number 1 here, and there it is. And just like before, audience members, just please go ahead and cast your vote. Remember, this is done in real time, so I'll be able to share the results with you once I close the polls. And the question we have for you here is, what applications is driving your interest in spatial transcriptomics? Okay, so you have a couple options below: immuno-oncology, cancer biology, infectious disease, therapeutic effects, or other. So please just go ahead and select one of the below, and then I will close the polls and we'll share the results and then get right back to Lester. Okay, perfect. Thank you everyone for participating in that poll question. I'm now going to close the polls. Okay, so let's take a look at the results. So we have here 17% are saying A, immuno-oncology, 39% are saying cancer biology, 22% are saying infectious disease, then we have 6% at therapeutic effects, and then 17% are saying other. So thank you everyone for participating in that poll question. Now back to Lester.
Well, thank you very much. Good news is I think we'll have examples that touch on all these interests in the presentation. So let's get going. All right, our first example in the overview starts with this lovely image from the review below, which is an excellent review I can recommend to anyone interested. And it highlights what we were talking about in the definition. In the left panel, you see the actual H&E, hematoxylin and eosin stained tissue section that began the study, where different areas of subtypes of breast cancer have been outlined for our convenience with the marker. In situ here, infiltrating carcinoma here, etc. And by applying spatial transcriptomics, we learn from gene expression that there are different patterns that cluster, and the clustering occurs even within histologically similar areas of tumor, emphasizing the heterogeneity of tumor and the heterogeneity of their biology, even within this very limited area. And this clustering can be further analyzed even at the gene level. Here's a cytochrome gene that is of some interest in cancer, breast cancer biology, which was analyzed throughout this grid. And you can see there's high and low and medium areas of expression, once again, including heterogeneity within apparently similar areas of tumor. And this is part of the promise of spatial transcriptomics, that is to say, to help us see through areas that may look similar histologically, but in fact have different biology going on with potential for different approaches to prognosis or therapeutics. I want to start with reminding ourselves that in fact, even though I'll be concentrating on one subset of spatial transcriptomics, this is a big tent with lots of different methods. Again, paraphrasing and taking from that review that's at the bottom, we can identify 3 major classes. There are even proteomic spatial omics, no longer transcriptomics since it's protein, and some of the methods are listed here. Fluorescence in situ hybridization methods are tabulated in the middle, and sequence-based methods for spatial transcriptomics are listed on the far right. And that's where the nanostring GeoMx method falls in. That is to say, it focuses on messenger RNA, although the same methodology can actually be applied to measure proteins, as I'll mention along the way. Let's look at a couple of examples of this platform in action. This is from the paper cited below, which is the sort of flagship paper describing the methodology from Nanostring. It's a wonderfully detailed presentation of the methodology. I have to warn you, it weighs in with the supplements at around 60 pages of dense data, but it does a good job of presenting the case and describing the methodology. Here they have studied colorectal cancer of 3 categories: microsatellite stability or instability, MSS, MSI, respectively, and whether or not the tumor inflammation signature graded separately on histology is either high or low. And to compare different areas of tumor, they pick regions of interest, shown here as these circles in high power in the panels to the right. And sometimes they study the middle of the tumor center that happens to have lots of immune cells and other areas, etc. And these are the kinds of regions of interest that the platform allows investigators to interrogate and compare across different types of tumors. And in this study, on the far left, we see a heat map of the gene expression levels across 3 kinds of tumor. And each tumor is divided into 3 regions: tumor center with immune-rich areas, or the invasive margin. And you can see there are quite distinct differences in the replicates, and sometimes having lots of gene expression of extracellular matrix associated proteins associated with the tumor center, and many other distinctions, which I won't go through. They may be perhaps better summarized, at least for our purposes here, to show the power of the technique with the heat map at the pathway level that they also offer. Once again, we have the 3 types of tumor with satellite instability and lots of inflammation, or satellite stability and high or low inflammation. And we can see clear differences in a variety of immune response pathways or categories. And this is the kind of analysis of different kinds of tumor biology that many people are interested in making progress with using this approach. Here's another example I want to share, which highlights the ability to study relatively small and precious tissue samples. This is a study of needle biopsies of kidney from patients undergoing acute kidney rejection. And you can see that samples are quite small. And these investigators compared glomerular versus tubulo-interstitial areas to see if they could learn and identify better genes to correlate with acute rejection for prognosis and detection. And in fact, in the volcano map on the right, they do show the right-hand side is the tubulo-interstitial area, a large panel of genes that they found quite interesting and will be part of their future investigations to achieve their goal of better markers of this process. The method has been applied to breast cancer and immuno-oncology. There was some specific interest in that in the poll. In this study, investigators looked at a aggressive triple-negative breast cancer and found that a subset of them, not a lot, a few, had detectable PD-L1, a marker of an immune response regulatory phenomenon in cancers, identified by immunohistochemistry, as shown as the little brown spots on this image here. And by selecting regions of interest from these kinds of tumors, as well as ones that don't have PD-L1 expression, they were able to further probe the immune response to the tumor. That's summarized in the heat map on the right. The PD-L1 positive tumors, when interrogated using these kinds of areas, as shown in C and D, have increased expression of a bunch of genes. And what are those genes? Well, it turns out, perhaps not surprisingly, there are many of them that are in fact other immune response genes, a lot of cluster of differentiation genes, markers of T cells, etc., and activation. And they take this to this conclusion, which I think is the spirit of the whole enterprise, spatially defined PDL1 tumor microenvironments were enriched in several clinically actionable immune proteins. And so these data may inform future studies on combinatorial immunotherapy along with standard PDL1 therapies already available. That's my wording at the end, but I think you get the drift of how this is impactful for these scientists. All right, and the final example before getting into specifics of the Nanostring program at Cellecta is one that instead of looking at RNA, looks at protein. These investigators were studying metastatic prostate cancer, which comes in two forms illustrated at the top. There's the lytic or bone destructive type. And in this panel at the upper left, you can see a sort of a snake of just pink tissue. That's the remnant bone, and it's surrounded by islands of tumor cells and stroma that are eroding the bone, hence it's and lysing it, hence the lytic designation. In contrast, there's another subtype, also aggressive, which is blastic, or which is a term used for bone-forming tumor. And here, the pink part of the tissue, which is a new bone, dominates the picture, and there are some islands of tumor triggering the formation of this bone entrapped in the bone. These investigators used antibodies with attached DNA barcodes to allow their quantitation by next generation sequencing to interrogate specific regions of interest, such as these islands of tumor in either lytic or blastic type of metastatic prostate cancer. Using this technique, they were able to quantitate expression of genes and found important differences between the two subtypes of tumor, the blue being the blastic, and they found PDL1 and other potentially targetable antigens quantified with this methodology. All right, I did want to, as we go, and here as well, talk about some of the limitations of spatial transcriptomics. I tried to share a few examples of successful application a moment ago. First, let's agree that RNA expression is not equal to protein expression. This is a concept that's bedeviled the field. Broadly speaking, depending on the review you look at and the criteria, probably 70 to 80% of RNA expression is matched by protein expression. If the RNA goes up, the protein goes up, and vice versa. But there are important exceptions, and they vary from tissue and model. So you can't come up with a specific single number, but we should be aware that there can be cases where the two are disconnected. The sensitivity for very low copy number messenger RNAs is not established. There hasn't been a really good benchmark study across multiple tissues, so that remains to be an open question. MicroRNAs, long non-coding RNAs are generally not covered. Mostly the platforms focus on the protein coding common genes, which is plenty of information, but nevertheless, we should be aware that this is not covered. The throughput is best described as medium, certainly not high, because it's at this per slide, per assay level. I'll mention some numbers for the GeoMx in a moment, but it's not a super high throughput methodology. The statistical analysis is evolving, and it sounds like a bit of a dodgy term. There's certainly very good methods in use that provide good answers. Nevertheless, when you get under the hood, there is some discussion in the literature about what the proper normalization methods are. They may vary depending on the size and number of regions and the statistical models used. So this is an evolving area. Now we're going to be heading more towards detail of the GeoMx nanostring platform we offer at Cellecta. Let's walk through what happens in an assay. We get a paraffin embedded slide or cryostat section fixed slide. The initial step is to stain the slide with the probes, and the probes are targeting specific messenger RNAs in the tissue, and they come with attached photocleavable DNA sequences. Once the labeling is done, we can image the slide, and so the staining includes the invisible probes of RNA, but also immunofluorescent probes to identify areas of tissue. More examples of that coming up. We can image the slide and then select our regions of interest based on the specific experimental questions being asked. Here's where the technology really takes off. Built in is an array of about 1 million 1 micron squared micromirrors that can be controlled to flash UV light at a very, very targeted area, namely the ones you have chosen as the regions of interest you want to sample. After that flash, the photo-cleaved oligos are available and they are aspirated quick like a bunny by a microcapillary. And then the system moves on to the next region of interest. The aspirated samples are individually placed into 96 well plates that are coded to match the ROI and the region that you have selected. And eventually this plate gets turned into a library for NGS readout on an Illumina sequencer. Let's go into some more details. There's panels available for either the whole transcriptome or a targeted panel for cancer, called the Cancer Atlas, about 1,800 genes, about half of which are clearly oncology related, and the other half are immuno-oncology related, since the two are travel together in interest in investigations. You can profile regions of interest of almost any shape using this technology. There is a minimum recommended region size of 5 by 5 micron. However, the recommended minimum in practical terms is somewhere around 50 to 100 microns squared. The maximum is listed there. You can get actually quite big. Usually there we use ROI as at least half as small as that, and that's sufficient. You can select subsets within regions of interest by segmentation or masking. For example, you can ask for collection only from the pan-cytokeratin positive tumor cells or from the CD45 positive leukocytes, etc. There are 4 fluorescence channels. I'll have some more information about how those are used later on. Either paraffin embedded or cryostat sections are okay for this methodology. And interestingly, you can reprofile the same section. All the probes can be washed off. It's non-destructive, so you can go look at other areas with other probes later on. As I mentioned, the throughput is medium per experimental run. You can use from anywhere from 1 to 4 slides. The shapes and options for regions of interest are illustrated here. They include the images on the top are the target, and below we see the kinds of shapes you can select. They can be geometric, and here we have a circle, but in fact they can be a square or a polygon. You can segment or mask different areas. Here the segment has been chosen to focus on the cytokeratin positive tumor cells in this sample, and so that would be the area that gets the UV flash and is collected. Similarly, you can look at other cells. Here, the tumor cells are ignored, and the mask is to collect all the CD68 positive macrophage materials. So you can collect, in a sense, from single cells. This is not a single cell technology because all the information from this subtype of cells will be pooled, but what you're going to get is information about the pool of the cells, say, near a tumor versus away from a tumor, etc. You can also select other unusual shapes. The contour is illustrated here. You can mask more than one type in a single section, as shown here. This illustrates this concept of different ROIs and how they work in a little more detail. In this example, Nanostring has shown labeling of tonsil tissue with DNA, CD3, or pan-CK in 3 different colors. And then they've performed a gridded regions of interest collection across the tissue, as shown here. Now, this illustrates a couple of good points. First of all, if you label things with pan-cytokeratin in green, you would hope that the gene expression would include high expression of keratin genes, and in fact, that is the case, as shown here. This is the keratin expression, and more or less corresponds to what we see by immunofluorescence for pan-cytokeratin. Similarly, areas that are high in CD3 positive leukocytes have high CD3 expression at the gene level, as shown here. It does also remind us that no methodology is completely perfect, and you can see these gray zones. There are a couple of like this square here has no keratin detected, but this clearly is cytokeratin here. And whether this is an edge effect or part of some stochastic process is unclear. This is part of the reason the general practice is for any given query, we collect 3 or 5 different regions of interest that are replicates to pool the data subsequently and to avoid any potential impact from the occasional outlier. And here's another one where there's plenty of CD3 staining, but not much signal once we get to the transcriptome level. All right, so some more details and facts about the platform. Broadly speaking, the minimum number of cells in any region of interest should be at least 50 to 100, depending on the cell size and their RNA content. There are occasions where you can get down a little bit lower to say 30, but that's a useful guide. As I mentioned, the minimum number of replicate ROIs should be somewhere at least 3, and 4 or 5 is better. Across any study, we can keep try to keep the ROI size and cell numbers consistent to make comparisons more uniform. Excuse me. We avoid any tissue defects, any folds in the paraffin section, et cetera, or edge areas. Now, for sequencing depth and capacity, I offer here a couple of guides from this excellent review of best practices for spatial profiling, focusing on breast cancer research, but has a lot of good points for any interest in this technology. However, these are kind of, I realized reviewing the slides, these are kind of difficult to process. And there are algorithms and equations basically that Nanostring provides that help guide you figure out this question, how much read depth do I need? The one rule of thumb that I can leave you with is, for example, for 100 wells, 100 areas of interest, each of which is a 300 micron circle, which is pretty generous, they would recommend roughly 200 million total reads. Now, you can often use smaller ROIs and fewer AOIs, so the total reads required for that would be lower. And of course, if you use more, you would need more. All right, let's do a couple of use cases of spatial transcriptomics at Cellecta. The first one I'd like to share relates to tuberculosis. And so this might be of interest for the folks who clicked infectious disease in the poll earlier. Tuberculosis is one of the most common and serious infections throughout the world. The good news is 90% of people who get it are actually asymptomatic. They become TB skin test positive, but really does not cause any further problems. The real problem is about 10% of people do develop active TB, so-called secondary or progressive primary pneumonia. And because the denominator of infections is so huge, this 10% is also huge and a major clinical problem throughout the world. Dr. Kramnik at Boston University has been studying this, and he's developed some ideas and hypotheses on why this happens, primarily using exquisitely detailed in vitro mechanism analyses, but he wanted to move into in vivo. His overall theory is summarized in this graph, where there's a role for tumor necrosis factor, creating a vicious cycle of activation of NF-kappa B, type 1 interferons that inhibit antioxidant defenses, and this modulates even further production of TNF, et cetera, when confronted with mycobacteria, leading to a late progression when this occurs in individuals infected. Now, he wanted to test this, and the timing was good because midsummer, Nanostring released its whole mouse transcriptome atlas. This is an RNA probe set that targets about 21,000 transcripts for mouse protein coding genes, plus some important negative controls. The advantages of targeted probing includes that you can exclude uninformative high expression targets such as ribosomal subunits. And this is read out using Illumina next generation sequencing. The graph on the right shows the number of genes roughly that they can detect using different size diameter regions of interest. Even with the smallest one of 50, they can detect around 5,000 or more genes, either in paraffin embedded sections or fixed frozen sections. As you increase the region of interest, the number goes up to about 10,000 or so, which is a very generous sample and consistent with likely expression of a subset of the full genome in any given tissue. This example also allows me to show you something a little bit more about how the actual slide prep works. The GeoMx instrument can interrogate the area on a slide that's in green on the image on the left. There's a gasket that sits on top, which is the dark blue. So you can't use the whole slide, and you need whatever you're interested in to fall into this area. With Dr. Kramnick's project, we wanted to study a number of mouse lungs efficiently in one slide, and we tried to get 5 into this box. The histotechnologist did a great job of placing these sections from individual blocks into the target area. This one was off and was not studied, but it was only had one lesion and did not prevent us from making progress with the project. But this is sort of what you would like to try to have, the area where you'd like to have your section or sections of interest. So in this study with Dr. Kramnick, we looked at samples from early lesions and late to test his hypotheses. And here's an example of how the lung section looks on H&E, the corresponding immunofluorescently labeled section. And here's an example of an ROI we looked at. And the upshot of the study was that we found strong differences between expression in late lesions and early lesions. Early being about 3 weeks, this is being closer to 3 months. And the good news was that here's his original hypothesis. Once we looked at the differentially expressed genes, especially at the pathway level, the number one pathway was in fact TNF-alpha signaling, consistent with his hypothesis. There are a number of other mechanistic angles to pursue as a result of this that he was happy to see, including the second one, epithelial mesenchymal transition, which is a growing area of interest in the TB research area. Here's the second use case. This involves a study with a biotech company looking at cancer immunotherapy with a viral agent. Their goal is to enhance innate and adaptive tumor immune response to tumor. They were sampling from a clinical trial in patients. On day 0, a small biopsy. They started the viral injections, sampled again on day 3, and then had surgical removal of these tumors at day 21. And their goal in the spatial transcriptomics assay was to look for evidence of immune activation in the tumor or stromal area immune cells. Here's an example of the kind of analyses we did in this project. Here's on the left is a medium power view of the tumor in blue-green with cytokeratin, islands of adenocarcinoma. And in between is red labeling of lymphocytes with anti-CD8, the kind of immune cells they're hoping to enhance with their agent. This area can be broken into 2 masks or segments, looking at the tumor itself or the lymphocytes, which we did. And helpfully enough, in this study, they had clinical information allowing the samples to be classified as responders or non-responders. And looking at CD8 expression in these samples, we were able to classify the gene expression according to this and look at it by principal component analysis. And we saw a very good distinction between the 2 clinical subtypes using genes differentially expressed at a Q value of 0.05 or less with a fold change of 2-fold or more. And here's a heat map. It's an incomplete one of the different genes identified using this approach. You can see there's a difference between responders and non-responders. You can also see that, as with almost all real-world trials, not every patient falls perfectly into where you might expect. There's one non-responder who has a little bit of some of the message that characterizes the responder. Nevertheless, if we look at these genes and analyze them either by gene ontology or pathways, as in these 2 compilations, the top ones really speak to the kind of activation and lymphocyte influx that were of interest to the investigators. And you can see at the pathway level, interferon gamma response and inflammatory response are highly enriched in the responders in these CD8 selected areas. Now, at Cellecta, I want to acknowledge that it's a team of individuals that brings a lot of expertise to our service. You've been hearing from me. I'm a pathologist. We have Tianbin, Dongfang, Michael, and Nadia, who also bring expertise in other relevant fields that I've tabulated below. So there's a whole group of us that help escort the samples from start to finish as part of our analysis. And I want to detail that for you right now. This schematic is from Nanostring, and their version of an outline includes getting the FFP samples, pick a paneling profile, pick a morphology stain, select your regions of interest, get your data. It's more complicated than that, and I'll detail some of that, not overly detailed, but a little bit more for your interest. So the start begins with extensive project planning. As you might imagine from what we've been talking about, the experimental design is crucial, and we need to identify the specific questions to be addressed, because that drives choices about the number of sections, the number of slides, what are going to be the control areas, how many replicates do we need of what kind of region of interest. The middle of the process is the actual assay. Now, we've primarily been working with paraffin embedded sections. It's relatively important that these be recently cut, let's say less than 2 to 3 months old. And ideally, it would be nice to have matching H&E sections to guide the future ROI selection once we get into the immunofluorescent stained imaging within the instrumentation. Any antibodies that are going to be tested on an investigator's sections, we want to validate that they work as well as they should in a mock assay before the real assay happens. The assay is, after we label the slides, we do a basically live region of interest selection with remote video contact with the investigator, who can see the images as we see them in the machine, and can either approve our selection of ROIs, make suggestions, or in some cases we had an investigator who was familiar with the instrumentation and they could take over the mouse and drive the ROI selection process themselves. This is followed by library preparation and sequencing with quality control evaluation of the samples along these steps. I want to break in the description of the protocol briefly for 2 points. First is why the 2 to 3 month age limit on paraffin embedded sections. This is primarily based on Nanostring guidance, and the data that they offer that is behind this guidance is the single images shown on this slide. Namely, they have compared tonsil tissue, and they either have freshly cut or 2-week-old or freshly cut versus 3-month-old sections that they just let age for 2 weeks or 3 months. And they compared the total counts in multiple replicates of ROIs, and you can see they have very good concordance at 2 weeks, and it drops off a little bit at 3 months. It's not clear to the extent that this little drop off, how significant it is, how many genes are lost, or is it just all the same genes are detected with slightly lower amplitude. The details are not available. I've asked but have not succeeded in getting them. But I think it's a useful rule of thumb that we ought to try to use the sections to when they've been cut as possible. Anecdotally, we have noticed some drop off and some signal in paraffin embedded sections that are a bit older, but really that's anecdotal and not rigorous. The second bit of data I want to share with now is the part about labeling the slide to identify morphologic areas of interest, or so-called morphology markers or immunofluorescent labels. The instrument comes with 4 channels. Typical dyes are FITSI, Cy3, Texas Red, and Cy5. 1 channel is reserved for DNA for nuclear stain, and this is mandatory. It's what the instrument uses for focus. Then 3 are available for different antigens, typically pancytokeratin and leukocyte common for tumor studies. CD8 can be added as a third, as an example. There's an example that I shared with you, but These three are really up to the investigator. You can choose from their evaluated markers. For the humans, there's about 60 human markers, and for mice, there's about 20 that have been validated to work on paraffin-embedded sections. Now, these will have to be revalidated on your section of interest in-house, because we want to make sure that everything's working as planned before we do the final run. Now, in addition to immunofluorescence guidance of the RNA transcriptome profiling, I did mention that it is now possible to use oligo-conjugated antibodies to quantify antigens in multiplex form. And there's about 85 of those kind of antibodies, anti-mouse, about 140 anti-human, that are tabulated by Nanostring as validated and okay to use for that methodology. And they offer panels of 10, so you can look at 10, 20, or 40 different antibodies and quantitate them by next generation sequencing, as was illustrated in one of the examples I mentioned earlier. All right, time for our last exciting poll question. Sonia, can you take over, please?
Thank you very much. I was just letting the audience know we're going to run one last poll question, and we'll try to squeeze in maybe one question if we can. Let's run that poll question right now, and there it is. The question is, how interested are you in multiplex spatial profiling of antigens using panels of oligo-tagged antibodies? So please select one of the below: not interested, somewhat interested, or very interested. So please go ahead. Okay, thank you, everyone. I'm just going to close the polls now. Please just go ahead and select one of the below. We'll close the polls and share the results and get to one Q&A question. Okay, and here we go. We're going to close the polls and share the results. Okay, so the results are here. We got 15% are saying not interested. We have 62% that are saying somewhat interested, and then another 23% saying very interested. So thank you, everyone, for that. And now back to Lester, and then we'll get to the Q&A once he's done.
Thank you very much. Yeah, thanks for answering that. We have about 1 minute left of my presentation, so thank you for your patience and interest. The final part of our process is, of course, the data. And we use the GeoMx software, which is quite robust, to in a workflow that includes quality control, background correction, normalization, analysis of differentially expressed genes and pathways. And we give you a report with raw and processed data files in case you want to try your own bioinformatics approaches to the same data. We also have additional expertise available as needed if the situation arises. So the final strength I want to emphasize is this quote from a paper about users' perspective on the GeoMx platform. In our experience, defining good ROIs requires strong guidance from an experienced pathologist. That would be me in the case of Cellecta, but if you engage any other service, please try to get a pathologist involved. It really does help with getting the best results from the tissue. So in summary, we had an overview of spatial transcriptomics, which does fulfill these goals as in our definition of looking at specific targeted areas in tumor or disease sections. We talked about ways to try to get optimal results that include good design and planning and consideration of the technical requirements of the platform. And we have tried to share with you some of the best practices and expertise available at our service at Cellecta. And so thank you very much. I want to acknowledge all the members of our team as well as Dr. Chenchik at Cellecta. Thank you. Sonia, please take it away.
All right, thank you very much for that very engaging and very detailed presentation. I hope everyone enjoyed it. Now let's get into a couple of questions before we end the webinar. So if I can get Lester and Paul to put on your webcams, and away we go. Okay, and here we are. Hi, everyone. Okay, here's our first question from our audience member. This audience member is asking, how long does spatial transcriptomic project take from start to finish? So how long does it take? Who would like to answer that one?
I'll go. It's pretty easy to answer. Assuming everything works well with the validation of the antibodies, et cetera, roughly 3 to 4 weeks from receipt of the sections to data and report.
Okay, perfect. Let's squeeze another one in. Can you add custom probes to the panels? This audience member would like to know.
Yes, indeed. And in fact, there's a workflow at Nanostring where up to about 30 total custom probes. That's the maximum. We've done up to 5 or 6 in reality, can be ordered and included into the panel. So that's a definite yes.
Okay. You know, since we're doing so great with going through these questions, let me just add one more. Is that okay with everyone? Paul and Lester, good? All right.
Sure.
All right, here's our next one. Are there negative control probes in the panels?
Oh, absolutely. That's part of the way the quality control is assured, et cetera. And so there's at least 10, I believe, possibly more, and depending on the panel. So the short answer is yes, indeed. For the detailed number, I would have to look it up, but I think that may be good enough for the questioner.
Okay, yes. And here's the next one, last one for sure. What is the maximum number of sections per slide?
It's in our hands so far, as I showed you, we've maxed out at 4. There's one illustration from Nanostring where they seem to have about 6 or 7 tissues on a single slide. I think that might be the theoretical limit, but I think practically speaking, depending on the size, somewhere between 2 and 4.
Okay, perfect. Thank you so much, Lester, for going through some of those questions with us. We appreciate that. Well, thank you very much for those questions. We have reached the end of the Q&A portion of the webinar. If we couldn't attend to your questions, the team at Cellecta will follow up with you after this presentation, and there are quite a number of questions there that they will follow up with. And if you have any further questions, please direct them to the email address that's on your screen, and there it is, info@cellecta.com, or reach out to them by calling 1-877-938-3910, or check them out at cellecta.com.